So today I will be presenting some work that has been going on for quite some time. So I'm very excited to be presenting this, where there is this project I'd like to call the Gut Microbiome Tree of Life project. And the main motivation for this project was basically we wanted to understand how gut microbiomes could be better understood in the context of animal evolution. This has been an ongoing question. There's been many studies that have tried to answer this, but one thing that was lacking especially when thinking of going from something like the first gut that might have evolved 1 billion years ago, all the way down to the most recent mammals that evolved, is we really lack a sort of comprehensive database to even try to tackle this type of question. And so that was kind of what led to the Gut Microbiome Tree of Life project, which I started during COVID lockdowns, actually, because I was looking for something to do that was completely dry lab. And I'm forever grateful for finding this, because now I don't, I realized I didn't like wet lab work so much. But fast forwarding through, I guess 4 years later, we compiled all these different data sets from different sources. We stuck to 16S because shotgun data just there's not enough of it to cover the species that we want to cover. So we stuck with 16S, and we are eventually led to a data set with more than 20,000 samples across almost 300 studies, spanning 1,500 host species and covering 7 phyla across the animal kingdom. And the main goal here was really we were really trying to beef up numbers that had traditionally been underrepresented, especially insects. For example, you can see we got a lot of insects, but even just the other invertebrates that are a lot more rare in the literature, bringing those into this comparative analysis was critical, because up until then, you really just do not see these in comparative analysis at all. Most of the time, it's an analysis just on mammals, or maybe mammals and a couple of other vertebrates, but not vertebrates and invertebrates spanning the entire tree of life. And so fast forwarding a bit through basically, I guess, 4 years of many blood, sweat, and tears of harmonizing data and harmonizing a ton of metadata that we had to put together, we end up with this really nice data set. So here I'm showing a data set mapped across a host phylogeny. So it is collapsed at the host species level. So every tip you see a bar plot showing that host's gut microbial diversity. And what I would like you guys to focus on is the sea of red that you see at the top. This is mostly Pseudomonadota. And we see this stark transition that starts to happen around amphibians and the rest of terrestrial vertebrates. And it's basically this kind of explosion of Bacteroidota and then Bacillota, two different clades of Bacillota. And so this is a really stark shift, and it definitely raises a lot of questions as to why this is happening. And so one analysis that we did to just start the sort of follow-up analysis on this type of stuff was an ancestral state reconstruction. And if there's any evolutionary biologists in the crowd, you might be scratching your heads here. I know there's some problems with this analysis. We're making a major assumption here that a gut microbiome can be considered an evolutionary trait, but there is literature backing this up that this analysis is quite useful, given how phylogenetically linked gut microbiomes are when you look at hosts at a broad scale. And what we found was that on the left side, we have the analysis done at the microbial phyla level. And then on the right side, we did it at the order level. And what we found was, if we're looking at ancestral states, initially, it seems like the first quote unquote gut that might have existed was likely dominated by Pseudomonadota, and in particular Enterobacterales species. And then it wasn't until around 350 million years ago, so this is a bit close to the 365 million time point that tetrapods evolved, right? We then see this stark shift towards Bacteroidota and then eventually Bacillota. And so this analysis in no way, shape, or form sort of provides these like conclusive sort of conclusions about like gut microbiome evolution in any way. But I think what it does is provides everyone with this really nice sort of baseline time points that we can use for further testing, particularly in getting long read sequencing or shotgun data would really help add some further testing to these baseline time points. Another interesting thing that we could do is we can plot all of our data onto a PCOA plot. So every dot that you see here is a gut microbiome sample. And one thing that we did was we added in environmental samples as sort of this kind of like out group to compare to our gut microbiome data. And all of the environmental samples really clustered tightly together, even though they were coming from a bunch of different sources from around the globe. And as you move away from the environmental samples, you start to see more and more clusters of vertebrates. Whereas if you move closer, you start to see more and more clusters of invertebrates. And so one thing I would like to propose here is that it does seem to be that there is this sort of functional cluster of terrestrial vertebrates that we see that mostly show up on the left side. And so just to note that these two plots are exactly the same as you're seeing on the left, just they're a density plot. So they're highlighting samples by their density. And on the right side, we have environmental samples highlighted along with invertebrates and fish. And so it seems like these are two different distinct guilds, although obviously that's a problematic word to use here, but there is definitely something going on between these two. Another thing that you can see is, so if you plot the Bacillota over Pseudomonadota ratio as some kind of phylogenetic biomarker, you can see that all of our hosts do line up quite nicely with this ratio, where we have all of the vertebrates on the left, and then we have all the invertebrates on the right, minus some exceptions like sharks and lampreys. And then smack in the middle is our environmental samples. And so we do think that there is this sort of biologically governing rule, if you will, about animal gut microbiomes across the tree of life. And this ratio seems to be like a reliable biomarker to be able to predict if something is a vertebrate or an invertebrate or an environmental sample. So we think that could be useful for researchers going into the future. And then another cool thing that you could do with this data set is you could take microbes of interest that you might have. And this is cool because you could literally be a human health researcher, right? And you're interested in some human health microbes that have been studied, like Akkermansia, Mucinifila, or fecal bacterium prosnitzi. And you can just be like, oh, I wonder what they look like across the tree of life. Maybe this could give me some evolutionary context for the microbe that I'm studying in this mouse model, for example. And so you could very much just go into our data set and see its diversity across the tree of life and see if you could make any sort of additional hypotheses based off this new knowledge. I think could be very powerful for a lot of researchers. Similarly, you can be interested in Wolbachia. Wolbachia is very well studied among insects. And here you can actually see Wolbachia diversity across insects quite nicely. And I actually don't think this has been done at this scale yet. And so I really am actually interested in collaborating with anyone that works on insect gut microbiomes, because I think there's something cool to build here where we could expand this tree to even more insects and do some really cool stuff. So yeah, but I guess, so just going big picture here, the data set definitely doesn't make any like conclusive sort of claims about what gut microbiomes look like millions and millions of years ago. I think we can only just guess, and I think this is just a great starting point. But from what I can take from this data set and what I want people to take away from this is that it seems like the environment largely played a major role during animal evolution, especially during early evolution. And then it wasn't until you really get towards fish, the first vertebrates, that you start to see the beginnings of host factors start to take shape, but not really until terrestrial vertebrates that you see it sort of outtake the environment, where you have things like complex gut morphologies evolving and more complex immune systems that are evolving, with mammals having the most complex immune system. And so I think those are the driving factors, and there's probably a lot more that we need to look at that end up with a system where you have animals actually selectively recruiting for things like Bacteroidota or Bacillota, which I have in the old name here, versus Pseudomonadota, where we just see less and less of it in our vertebrate samples. So yeah, that is, in a nutshell, that's the data set. There's a lot more that we did, which you can read about in the preprint. And also happy to take questions after as well. So I'll just lastly thank my mentor, Dr. Rob Knight, Dr. Katie Amato, the Knight Lab team, and then all the amazing Northwestern undergraduates that spent many, many, many hours helping me coalesce thousands of metadata files together, which very fun time. So yeah, thank you.